Good morning, friends. My topic is chromosome walking and jumping. Chromosome walking and jumping. Chromosome walking and chromosome jumping are two technical tools used in molecular biology for locating genes on the chromosome and physical mapping of the genomes. Chromosome walking and jumping are used for physical mapping of the genomes and also for locating genes on the chromosome. Chromosome walking is a technique used to clone a target gene in a genomic library by repeated isolation and cloning of adjacent clones of the genomic library. It can also be used to clone a target gene. Chromosome walking can also be used to clone a target gene in a genomic library. It is done by repeated isolation and cloning of the adjacent clone of the genomic library. Chromosomal jumping is a special version of chromosomal walking which overcome the breakpoints of chromosomal walking. So the disadvantages of chromosomal walking can be overcome by using chromosome jumping. Chromosomal walking can only sequence and map small length of chromosomes, while chromosomal jumping enables sequencing of large parts of the chromosome. So chromosomal walking can only sequence a small length of chromosome, but the chromosome jumping enables sequencing of large part of chromosomes. This is the main difference between chromosomal walking and chromosome jumping. Chromosome walking. It is a method of positional cloning used to find, isolate, and clone a particular allele in a gene library. Chromosome walking is a method of positional cloning used to find, isolate, and clone a particular allele in a gene library. Chromosome walking was developed by Welcome Bender, Per Sparer, and David S. Hockness in early 1980s. It was developed by Welcome Bender, Per Sparer, David S. Hockness in early 1980s. There are nearly half a dozen positional cloning tests that are done prior to a chromosome walk. Each clone in the cosmic library has a DNA insert of 50 kb. So each clone in a cosmic library will have a DNA insert of 50 kb. The walking starts at the closest gene that has already been identified, known as the marking gene. So where will the walking start? Chromosome walking will start at the closest gene that has already been identified, and that is called as marker gene. Once the markers on either side of an unmapped sequence are found, the chromosome walk can begin from one of the markers. So when the markers on each side are found, chromosome walk can begin from there. From either of the markers. Each successive gene in the sequence is tested repeatedly, known as overlap restrictions, and mapped for their precise location in the sequence. Each successive gene in the sequence is tested repeatedly, and it is known as overlap restrictions and mapped for their precise location in the sequence. Eventually, walking through these genes reaches a mutant gene in an unmapped sequence that bind to a fragment of a gene of that particular disease. For a particular disease, walking through genes reaches a mutant gene in an unmapped sequence. Then it will bind to a fragment of a gene of that particular disease. The testing on each sequence clone is complex, time consuming, and varied by species. This series of overlapping clones could, for example, consist of bacterial artificial chromosomes. Hybridization probe. A more straightforward approach thus is to use the insert DNA from the starting clone as a hybridization probe to screen all the other clones in the library. More straightforward approach is to insert DNA from the starting clone, and that DNA is called as a hybridization probe, and thereby we can screen all the other clones in the library. Positive hybridization signal that are given by clones whose insert overlap with the probe are used as new probes to continue the walk. So the walk can be continued by positive hybridization signals that are given by clones whose DNA fragments, that is the inserts overlapping with the probe, are then used as new probes for the continuation of the chromosome walk. There are about 96 clones that a library consists of, and each clone contains a different insert. There are 96 clones in a library. A probe may have a genome-wide repetition of sequences. This can be reduced by blocking the repeat sequence with pre-hybridization with unlabeled genomic DNA. A probe will have a wide repetition of sequences, and these repetitions can be reduced by blocking the repeat sequence with pre-hybridization with unlabeled genomic DNA. But this isn't that effective solution, especially in case when high capacity vectors such as BAC or YAC are used in the work. For chromosome work with human DNA, which have high rate of repetition, intact inserts are not used in general. We know human DNA have high rate of repetition. For chromosome working in human DNA, we will not use intact inserts. Instead, the probe is taken from the end of an insert which has a lesser chance of repetition. So, this can be overcome by taking a probe from the end of an insert that will consist of lesser chance of repetition. Here is a picture depicting the chromosome walking. Application. This technique can be used for the analysis of genetically transmitted diseases to look for mutations. So one application is for analysis of genetically transmitted diseases and to check for mutations. Other application is chromosome walking is used in the discovery of single nucleotide polymorphism of different organism. Single nucleotide polymorphism of different organism can be found out by chromosome walking, and it can also be used for analyzing genetically transmitted diseases and to look for mutations. Disadvantages. There is a limitation to speed of chromosome walking because of the small size of the fragment that are to be cloned. So the major disadvantage is that because of the small size of fragments being cloned, the speed of the chromosome walking will be limited. Another limitation is the difficulty of working through the repeated sequence that are scattered through the gene. We have already discussed about the difficulty of chromosome walking through repeated sequence. So the other limitation is the difficulty of walking through the repeated sequence that are scattered throughout the gene. If the markers were too far away, it simply was not a viable option. Additionally, chromosome walking could easily be stopped by unclonable sections of DNA. Chromosome walking can easily be stopped by unclonable sections of DNA. A solution to this problem was achieved with the advent of chromosome jumping, which allows the skipping of unclonable sections of DNA. So, chromosome jumping was invented to overcome the disadvantages of chromosome walking. Chromosome jumping. Chromosome jumping is a special type of chromosome walking that takes advantage of the breakpoint of chromosomal rearrangement as guidepost and permits the cloning of the two end of a DNA sequence without the middle section. So chromosome jumping is a special type of chromosome walking that will permit the cloning of the two end of a DNA sequence without the middle section being involved in it. The procedure may take advantage of existing chromosomal rearrangement, or the genomic DNA may be partially digested with any restriction endonuclease or with enzyme that cut very rarely. It takes the advantage of existing chromosomal rearrangement or the genomic DNA will be digested with any restriction endonuclease or with the, any other enzymes. In order to bring the molecular marker close to the gene of interest, chromosome jumping approach can be utilized. This technique can be described in the following step. The distance of jumps or hope size is determined. First of all, the distance of jumps or hope size is determined. Example 100 kb or 200 kb, such that genomic DNA molecules in the range of selected sizes selected through pulse field gel electrophoresis. So how is this being selected? Genomic DNA molecules in the range of selected sizes selected by pulse field gel electrophoresis. For circularization of DNA segments, a ligation between two ends of each long linear molecule is allowed using T4 ligase. So for circularization of DNA segments, T4 ligase is used and it is being circularized. These circular DNA circles are digested with echo R1. Then this circular DNA circles are digested using echo R1. Phage vector CH3A lag is also cut with echo R1 and used for cloning small DNA fragments. So for cloning small DNA fragment, phage vector such as CH3A lag is also cut along with echo R1. Such cloned DNA fragment represent the jumping library, which can be plated on a bacterial host and screened through the technique of plaque hybridization. So this cloned DNA fragments is representing the jumping library, and it can be plated on a bacterial host, and then it can be screened through the technique of plaque hybridization. Technique of chromosome jumping helps in narrowing the gap between the gene and available molecular markers. After several cycles of chromosome jumping followed by cloning the region proximal to the gene, it will be possible to approach very close to the desired gene. The gene for cystic fibrosis has recently been isolated using this technique. So, the technique of chromosome jumping will help us in narrowing the gap between the gene and available molecular markers. After several cycles of chromosome jumping followed by cloning the regions proximal to the gene, it will be possible to approach very close to the desired gene. We will get genes that are close to the desired gene. The gene for cystic fibrosis has recently been isolated using this technique. Chromosome walking is frequently applied when it is known that a particular gene is located near a previously cloned gene in a chromosome, and it is possible to identify it with repeated isolation of adjacent genomic clones from the genomic library. Chromosome walking is frequently applied when it is known that when we already knew that a particular gene is located near a previously cloned gene in a chromosome, and then it is possible to identify it with repeated isolation of adjacent genomic clones from the genomic library. However, when repetitive DNA regions are found during the chromosomal walking technique, the process cannot be continued. Hence, the technique breaks up from that point. Chromosome jumping is a molecular biological tool which overcomes this limitation for mapping genomes. It bypasses these repetitive DNA regions which are difficult to clone and helps in physical mapping of genomes. This is the main difference between chromosome walking and jumping. When repetitive DNA regions are found during the chromosome walking technique, it is a limitation for chromosome walking. Hence, that technique breaks from that point. Chromosome jumping is a molecular biological tool which overcomes the limitations of chromosome walking. It bypasses these repetitive DNA regions which are difficult to clone and help in physical mapping of genomes. And it is a main difference between chromosome walking and jumping.